And where we're headed to today is we're going to run through the idea of spinal cord structure and the tendon tap reflex, along with some ascending pathways. The way we're going to approach this is I introduced last time the rudiments of the tendon tap reflex, and we're going to repeat that today. But we'll start here with the cross-section of the spinal cord, trying to get in your mind's eye a vision of how the spinal cord is oriented and organized in cross-section. So we're going to draw basically a circle on the board and then put in gray matter and white matter and work our way through that. We'll use this opportunity in describing the structure of the spinal cord in cross-section to emphasize some orientation nomenclature or language. We're going to talk about the region of the spinal cord which is toward the back. This is said to be dorsal, more commonly these days referred to as posterior. There's then going to be a region toward the belly, which I typically refer to as ventral, but is more commonly referred to in humans particularly as anterior. Then we're going to divide the structure that we see in cross-section of the spinal cord into a region that's the central nervous system and a region that's the peripheral nervous system. We're basically going to put a dotted line around the spinal cord and say that we have an interface between that which is central and that which is peripheral. Within the central nervous system, we're going to find that there are regions called gray matter and regions called white matter. When you see the term gray matter, what should immediately come to mind is the concept of input regions. These are, in the old anatomic nomenclature, these will be the dendrites and the cell bodies. Now, within the gray matter, we're going to see, using our nomenclature learned up here in terms of the orientation, we're going to see a dorsal, sometimes known as posterior, and a ventral, sometimes known as anterior, horn. So when you see the term horn, immediately think central nervous system, gray matter. And then we'll recognize that in contrast to the gray matter, which is organized almost in a butterfly kind of shape, in the center of the spinal cord, outside of that is going to be the region of the white matter. Now, the white matter was named long, long ago, before we understood the cellular organization of the central nervous system. And what we'll find here is when we talk about white matter, what we really mean are conductal regions. These are the long-distance communicating parts of the neurons. And the reason that the white matter is white is because it is ensheathed by myelin laid down by oligodendrocytes. So it's white because it's fatty. And when you see white, you think conductal regions because these are the myelinated axons. Then once we progress from the central nervous system out into the periphery, we'll see now the nomenclature changes a little bit. We go from the idea of horns, which are central nervous system, to roots. Roots are extensions of the dorsal horn and ventral horn, respectively, but now the dorsal root is going to contain the axons, the conductor regions of primary sensory afferents. The dorsal root ganglion, which is going to be a part of the dorsal root, and again in the peripheral nervous system, the dorsal root ganglion is going to be a cluster of neuronal cell bodies, but in this case, these neuronal cell bodies are going to be all primary sensory afferents, carrying somatosensory information. I'll emphasize it here too, and we'll say it a little later, that normally when we talk about a ganglion, what we're referring to is a cluster of neuronal cell bodies in the peripheral nervous system. But when we talk about a dorsal root ganglion, we certainly are talking about neuronal cell bodies. We're not talking about input regions. In almost all cases, when we're talking about neuronal cell bodies, we're thinking about the input regions of those neurons. But in the dorsal root ganglion, we simply have the cell bodies of the primary sensory afferents carrying sensory information into the central nervous system. And we'll draw that out. We'll then see that on the ventral or anterior side of the spinal cord, extending into the periphery, will be the ventral root. Just as there's a dorsal root, there's a ventral root. And in both cases, these are going to contain clusters or groupings, bundles of conductal regions. Then there's some definitions we'll need to repeat as we work our way through this. And again, ganglion is a collection of neuronal cell bodies in the peripheral nervous system. A nucleus is a collection of neuronal cell bodies in the central nervous system. A nerve is a bundle of conductile processes in the peripheral nervous system. We use this term nerve all the time, and it has a particular meaning when we talk about neurobiology. A nerve specifically is a bundle of conductile processes in the peripheral nervous system, whereas a tract is a bundle of conductile processes in the central nervous system. And we'll work our way through that nomenclature and give examples of each of them. And then we're going to drill down and talk about a particular circuit. And we introduced this circuit a little bit last time. And this is going to be the so-called patellar tendon tap reflex. And the patellar tendon tap reflex is such that when we tap the patellar tendon, the patellar tendon tap reflex, and when we pull on this tendon, we stretch this tendon, we're actually going to be stretching the quadriceps muscle. When we stretch the quadriceps muscle, we're going to activate a primary sensory afferent that responds to a deformation of its input region. Just as we saw with those sensory neurons at the tip of our finger, we now are going to activate these neurons by stretching the muscle, the quadriceps muscle. When these turn on, they then propagate action potentials all the way into the central nervous system, all the way into the ventral horn. And in the ventral horn, in the central nervous system, in the spinal cord, we find the cell bodies of lower motor neurons. This is an unusual circuit in that it's a synapse directly from a primary sensory afferent onto a lower motor neuron. That lower motor neuron then, once activated, will go out and synapse the quadriceps muscle. So we'll walk our way through this reflex where we stretch the quadriceps, set up action potential in the primary sensory afferent. Those action potentials will release glutamate onto the lower motor neuron, and once that glutamate activates the lower motor neuron, then that will then send action potentials to the muscle at the neuromuscular synapse, will then activate the muscle. We're also going to see, though, that in this circuit, there's a branch of the primary sensory afferent. And a branch of the primary sensory afferent synapses onto a local inhibitory interneuron. The local inhibitory interneuron is confined to the gray matter of the spinal cord. But what it will do is it will respond, this inhibitory interneuron will respond by firing action potentials from activity in the primary sensory afferent. The primary sensory afferent synapses onto the inhibitory interneuron. It will generate action potentials in the inhibitory interneuron. The action potentials in the inhibitory interneuron then will release the neurotransmitter GABA, or in the spinal cord probably glycine, onto a lower motor neuron. And what we're going to see is that as we activate the quadriceps muscle through the tendon tap reflex, we almost simultaneously activate the inhibitory neuron, which turns off the lower motor neuron that innervates the antagonistic muscle. One of the ideas we won't have time to spend a whole lot on is that all of our muscles are laid out in antagonistic pairs. That is, there's a flexor and an extensor. So in our thigh, the anterior aspect is the quadriceps muscle. That's an extensor. But the posterior aspect contains the hamstring muscle, and that's a flexor. So that when I kick my foot out, I'm using the quadriceps muscle to kick out. But when I draw my heel up to my rump, when I do that, I'm using my hamstrings muscle to pull that foot up. So we'll see these muscles laid out in antagonistic pairs, and then we'll look at how that innervation is reflected through the inhibitory interneuron. And one of the ideas I want to emphasize here, one that I think I don't get across sometimes, is that the lower motor neurons always release an excitatory neurotransmitter onto the muscle. That's always acetylcholine. It always binds to nicotinic acetylcholine receptors to initiate the muscle contraction through the action potential mechanism. Yes?
The patellar tap is happening independent of the CNS then?
So the patellar tap is happening independent of the CNS? No. Because now some of this is going to revolve around the idea of what we determine to be the central nervous system. So the central nervous system will be the spinal cord and brain. Yes. And so this information is going to go from the periphery into the spinal cord, synapse onto neurons in the spinal cord. That information is going to be carried back out to the periphery. So the central nervous system is involved. But now, as you point out, this central nervous system is only spinal cord. And then that's an important idea here, is that the reflex arc is confined to the spinal cord. We can cut the spinal cord and still get a reflex arc without delivering that information to the brain. And so then once we have laid out this idea of the local reflex arc, then we'll have to carry that information up to the brain through the ascending pathways. So again, when you say central nervous system, recognize you mean brain and spinal cord.
Correct.
Okay. Anything else? So we'll follow those sensory pathways. Now, what I want to do here is describe the anatomy of the cross-section of the spinal cord. I'll begin this simply by drawing a circle. That's meant to be the spinal cord in cross-section. And I think the best way to envision this is to recognize if this were my spinal cord, the way you'd be looking at it would be as though you were looking at my spinal cord with my feet up here on the desk and my head behind the board. And then you would cut a section through my spinal cord, and you'd be looking at that end on, cross-section end on. When you do that, then we'll divide the spinal cord into these regions. This butterfly structure here, and I'll hash it in black, this butterfly region is said to be the gray matter. Whenever you see this term gray matter, what you should be thinking of, what you should recognize, is that the gray matter represents input regions and output regions of neurons. From this orientation, we can also talk a little bit about the language we use to describe this. So again, if you envision me with my feet on the desk, this region is going to be toward my back. This will be right. This will be left. In this direction is toward my back. We call this dorsal. The other name for dorsal that's more commonly used these days is posterior. But then from the midline, we can draw another arrow in this direction. And we can say everything below the midline is toward the belly. We term this toward the belly as ventral. Toward the belly is ventral or anterior. We can draw another line through here and distinguish that which is right from that which is left. Now, if I've drawn this well, which I haven't, but if I've drawn this well, what we see on the right, we see on the left. So that the bilateral symmetry that we recognize when we look in a mirror or we look at someone else, that bilateral symmetry is extended all the way into the central nervous system. And so we'll have a right side and a left side, and that which is on the right is recapitulated on the left in a kind of mirror image. So now we can think of a kind of interface here. And in the dotted red line here, what I'm trying to distinguish is the difference between that which is part of the central nervous system and that which is part of the peripheral nervous system. So everything on the inside of the line is going to be central, or CNS. Everything outside of the line is going to be peripheral, or PNS. We can further divide this gray matter with a little bit more nomenclature. And if you take an anatomy course, you'll find that we divide this way down into a number of different regions. But the region I want to tell you about here is the region up here called the dorsal or posterior horn. The horn that I'm showing you here is gray matter. The horn that I'm showing you here is in the central nervous system. It's inside the red dotted line. So this is the dorsal horn. Down here on this side is the so-called ventral or anterior horn. So the gray matter is neuronal cell bodies and output regions. It's the integration site here within the spinal cord. The gray matter can be divided into the dorsal horn, which is dorsal, and the ventral horn, which is ventral. And then this region out here, out beyond the gray matter, it's hashed in orange here. This region out here is said to be the white matter. The white matter is, in fact, conductal regions. Of neurons. The reason the white matter is white is because of the myelin produced by oligodendrocytes. What we're going to see here a little later is that these conductal regions of neurons in the central nervous system are located in regions called tracts. Tracts are bundles of conductile processes. In the central nervous system. And in the central nervous system, for the most part, we see these tracts or conductal regions in the white matter. But now we have divided the region of the spinal cord into the central portion into the white matter and the gray matter. But the gray matter up here actually extends out into the periphery. And this is true on both sides. And extends out into a region here, which is kind of a bulbous structure, called the dorsal or posterior root ganglion. The posterior root ganglion is found as an extension of this short segment here known as the dorsal root. The dorsal root ganglion, as the name implies, because we said that a ganglion is a collection of neuronal cell bodies in the periphery, this dorsal root ganglion contains the cell bodies of primary sensory afferents, the primary sensory afferents of the somatosensory system. So if we would look at one of these neuronal cell bodies here, this is meant to be a cell body, there's its nucleus. This neuron will extend a process into the central nervous system. But similarly, this neuronal cell body will have an extension that goes out into the periphery. This will go out into a spinal nerve. Now, down here, meanwhile, back here in this region, in the so-called ventral or anterior horn. Again, this is gray matter. So down here, we see neuronal cell bodies. Neuronal cell bodies of lower motor neurons. Well, let's take a moment and bring this back around to what we talked about on the three-box model of the nervous system the very first day. And that is this blue neuron. This blue neuron here is a primary sensory afferent. You remember that a sensory afferent is carrying information, carrying action potentials toward the central nervous system. So this is a proper primary sensory afferent carrying information about, carrying sensory information in from the body. Down here is a lower motor neuron that now extends a process out into the periphery. And this lower motor neuron can be thought of as a somatic efferent. So again, the circuit, we have a somatic afferent carrying information into the central nervous system and a somatic efferent carrying information away. These somatic efferents, these lower motor neurons, are destined to synapse onto muscle. On the upper panel, I'm going to change that view a little bit. And I'm going to describe in a very primitive way a longitudinal. Longitudinal view of the spinal cord. So the longitudinal view of the spinal cord is going to look a little like this. And right outside the spinal cord, and I won't draw them all in, right outside the spinal cord will be the 31 pairs of dorsal or posterior root ganglia. So we see this repeating pattern all along the length of the spinal cord. There are 31 pairs of these guys. What we drew above would show this neuronal cell body in the dorsal root ganglion sending a process out, or having a process in the structure here known as the spinal nerve. The spinal nerve contains not only these sensory afferents, but also contains lower motor neurons, which now enter the spinal nerve. So if we were to draw this in the cross-section, what we see in the cross-section is that these primary sensory afferents and the efferents will join one another. They will join one another out here someplace and bundle together in something known as the spinal nerve. And again, here's the nomenclature. When we talk about a nerve, we mean a bundle of neuronal conductile processes in the peripheral nervous system. Later we'll see that there'll be bundles of axons, conductile processes in the central nervous system. Those will be tracts. So tracts are bundles of conductile regions in the central nervous system. nerves or bundles of conductal regions in the peripheral nervous system. So that's the overall anatomy. Now, let's draw over here what's known as the patellar. Tendon tap. Reflex. So that's my rendition of a leg. The muscle that we're going to be interested in is the one located on the ventral or anterior aspect of the thigh. This muscle here is said to be the quadriceps. The quadriceps has on it a tendon, and that tendon attaches this little disc I'm going to show you here in green, which is meant to be the patella. We commonly think of the patella as the kneecap. The patella, in turn, is connected to the two bones of the lower limb, the fibula and the tibia. So that's the mechanical anatomy of the quadriceps linked to the patella, to the fibula and tibia. And we've all had this experience. We've all done this. We've all gone to the doctor's office, and we've sat on the sort of gurney there, and the physician or PA comes in, and they ask you to fold one leg over the other. And then they come out with this hammer, that triangular-shaped rubber hammer. And the doctor or whomever feels around just below your kneecap. And what he or she is looking for is a soft spot which represents the tendon, the patellar tendon, the tendon that links the kneecap, the patella, to the fibula and tibia. Then once the soft spot has been found, then the physician will take that hammer and whack you right in that soft spot. And what happens, when they do that, is when they hit you in that soft spot, your lower limb kicks up, your foot kicks up. And the reason for that is that when you strike the tendon, you stretch the tendon that's connected to the patella, that's then connected to the quadriceps muscle. When you stretch the quadriceps muscle, there are sensory neurons embedded in the quadriceps muscle. Those sensory neurons are contain stretch-activated channels. So when you stretch the muscle and you stretch the input region of that primary sensory afferent, you now open those stretch-activated channels. Sodium comes in. It produces a generator potential. If the generator potential is large enough, it activates all the voltage-gated channels of the trigger zone, and now you propagate action potentials from your quadriceps muscle in that primary sensory afferent all the way back into your spinal cord. Then in the spinal cord, that primary sensory afferent synapses directly onto a lower motor neuron that now goes out and innervates the quadriceps muscle. So the circuit now is to stretch the quadriceps muscle, initiate action potentials in the primary sensory afferent, communicate to the lower motor neuron, contract the quadriceps muscle, and because that's an extensor, your lower limb kicks up. So let's follow that now through the anatomy. In blue is the input region here of the primary sensory afferent. This is the stretch receptor. This stretch receptor then extends into the neuron, the conductor region of the neuron. And then here's the neuronal cell body. The neuronal cell body is located in the dorsal root ganglion, also known as the posterior root ganglion. Then, using what we have above, drawing the spinal cord and the butterfly arrangement of the gray matter, This blue primary sensory afferent now extends in through the so-called dorsal root into the spinal cord. Extends all the way in the gray matter in the spinal cord down into this region. Extend a little farther. Into, this is the output region. I don't want to put it over there. This is the output region of the primary sensory afferent. The neurotransmitter that's releasing is almost certainly glutamate. It forms a synapse directly onto this lower motor neuron. This lower motor neuron, located in the ventral horn, then sends a process out into the ventral root, joins the spinal nerve, and finds its way to the quadriceps muscle. So the pathway here is now to pull on the quadriceps muscle, stretch the input region of this primary sensory afferent, fire action potentials in the primary sensory afferent. When the action potentials reach the output region here, they'll open the voltage-gated calcium channels. Calcium comes in, binds the synaptotagmin. Then through the snare complex, releases neurotransmitter via exocytosis. The transmitter glutamate now binds to receptors on the lower motor neuron. And almost always in this circuit, there's so much glutamate release that we activate so many of these ligand-gated channels that we are guaranteed to fire an action potential in the lower motor neuron. That action potential now propagates out into the periphery, all the way down to the output region of the lower motor neuron, where it releases acetylcholine onto the muscle fiber. The acetylcholine opens ligand-gated ionotropic channels. Their opening leads to the opening of voltage-gated channels. That propagates an action potential on the muscle fiber that we'll talk about on Monday, causing the muscle to contract. So this is the circuit that I described sitting on the desk and hitting my kneecap, hitting the tendon attached to my kneecap. That circuit is simply this two-cell circuit. Now let's look at this in a couple of slides. Now the first slide is to show the electrical events at each one of those stages. And this is a good place to review the electrical events that we see. In other words, out here in the muscle, where we have the input region of the stretch-activated neuron, we would have stretch-activated channels out here. When we pull on the muscle, we open them. That will generate a depolarization. Sodium comes in through those stretch-activated channels. This is a generator potential. When we open those channels, if there are enough of them opened and they drive the membrane potential above threshold at the trigger zone, that will now initiate action potentials. Those all or nothing invariant signals then will propagate along the axon, along the conductor region, over the neuronal cell body located in the dorsal ganglion and into the central nervous system, and then finally in the ventral horn, the primary sensory afferent will release glutamate onto the lower motor neuron. That neurotransmitter now will bind to glutamate receptors, generate a synaptic potential. The synaptic potentials are large enough, if we have enough synapses activated, they will then trigger the opening of voltage-gated channels here at the trigger zone and now propagate these action potentials along the conductor region of the lower motor neuron. When the action potentials are along the output region of the lower motor neuron, they open calcium channels, go through the snare hypothesis, release acetylcholine, and initiate the process of contracting the quadriceps muscle. That's the physiological circuit. The anatomical circuit that I drew on the board is shown in a much prettier fashion here in this figure. But again, if you follow the pathway, here in blue is the input region of the primary sensory afferent. The primary sensory afferent has its neuronal cell body up here in the dorsal root ganglion. It then sends a process in to the gray matter of the spinal cord. The process that we followed was to take this process and watch its synapse onto this green lower motor neuron. The green lower motor neuron then will send its process out to the quadriceps muscle. And electrically what's happening is that when we tug on this input region and generate action potentials, the action potentials release neurotransmitter at this site and cause the lower motor neuron to fire action potentials, causing the quadriceps muscle to contract through the release of neurotransmitter and the activation of the muscle cells. But there's another important circuit I want to show you from this figure. You'll notice the way this has been drawn, that this blue primary sensory afferent has a branch up here in the gray matter. And this branch now synapses onto this red neuron. This red neuron is meant to signify an inhibitory interneuron. Now, let's follow the process here. Action potentials are propagated into the central nervous system at this output region of the primary sensory afferent. The neurotransmitter that will be released at this site will be glutamate. The glutamate binds to receptors on the inhibitory interneuron. That glutamate opens ligand-gated channels, generates an action potential. The action potential then propagates to the output region of the interneuron. When the action potential arrives at the output region of the interneuron, it now releases neurotransmitter. But now this neurotransmitter is going to be GABA, or perhaps glycine, here in the spinal cord. That GABA then is going to open chloride channels here in this lavender lower motor neuron. When it opens chloride channels, this inhibits this lower motor neuron. So now what's happening? Let's look to see what's happening. In the process of acting the quadriceps muscle through this primary sensory afferent to lower motor neuron, we simultaneously are turning off the antagonistic hamstring muscle. We're not turning it off by directly affecting this synapse. We're indirectly affecting the synapse by turning off the lower motor neuron that innervates the hamstring muscle. So in vertebrates, all of this inhibition occurs in the central nervous system, and that inhibition impinges on the lower motor neuron at the synapse between an inhibitory neuron and the lower motor neuron. Now, we like to think we turn one on and we turn the other off, but remember the way inhibition works is to modulate things up or down, so that when this green lower motor neuron is on, causing the quadriceps muscle to contract, it's now at the same time through this circuit releasing inhibitory neurotransmitter to turn off this lavender neuron, so we're less likely to activate the hamstrings muscle. So it's basically this reciprocal circuit where when we activate the quadriceps, we turn off the hamstring. We could follow the same pathway in the other direction, where if we turned on the hamstring, in other words, to cause a flexion of the hamstring and bring the foot up toward the rump, if we did that, then we'd be activating the hamstring, but through a similar circuit, turning off the quadriceps. Because these antagonistic muscles, when we activate one, we want to slow down the other, because we have flexors which pull the muscle on the joint to cause flexion, and we have extensors to extend the joint. So in this case, if you think the lower limb, when I activate the extensor, I'm kicking out. That's quadriceps. But if I want to draw my foot up, I'm activating the hamstrings, which is now activation of the flexor. Any questions to there? That's the reflex arc. And recognize that an important part of the reflex arc is that inhibition of the antagonistic muscle. Well, as we pointed out earlier from one of the questions, let's now imagine what happens if we simply cut the spinal cord here. We know if we cut the spinal cord, effectively, we'll no longer have any sensation in those regions below the cut. And we'll also know that we've lost ability to voluntarily activate regions below the cut in the cord. But even though we have cut the cord at this site, this circuit remains intact. So if you have a spinal cord injury, either in a human or in an animal, you'll find that the information from the periphery is not propagated to the brain because the spinal cord is cut. but you still retain the spinal cord reflex, because this is a local reflex. And in fact, over time, these reflexes get stronger. So in cases of spinal cord injury, if you do one of these tendon tap reflexes, this reflex gets stronger and stronger over time, mostly because of the loss of descending inhibition and some loss of local inhibition. But with that, what I want to do is begin thinking about how this information is going to go up the spinal cord and arrive at the brain. So on the right-hand side here, I want to spell out the pathway of activity. And we're going to make a list here, and then we'll try to follow it through on that ice cream cone diagram. And the list is going to be such that we have a primary sensory afferent. The cell body will be in the dorsal root ganglion. The primary sensory afferent synapses onto a second order neuron. The second order, I should say, the process, put in parentheses, conductile region, of the second order neuron crosses the midline. Then this second order neuron synapses onto a third order neuron in the thalamus. And the third order neuron, synapses onto neurons in the cerebral cortex. So let's heal this spinal cord. Let's follow this neuron. Again, let's put it back into the context of the quadriceps muscle. So this will be a sensory neuron with its input region in the quadriceps, just like we had before I erased it. This neuronal cell body is in the dorsal ganglion. It now enters the spinal cord. Now, the emphasis we had is how this primary sensory afferent here, in cross-section, would extend a process down into the ventral horn and synapse directly onto the lower motor neurons. That's the drawing we had. And that's true. But there's another branch of this primary sensory afferent. Another branch of this primary sensory afferent now enters the white matter. It enters the white matter in the dorsal part of the cord that has a name called the dorsal columns. So while it's true, as we showed in the cross-section, this neuron synapses onto the lower motor neuron, it also has another branch that enters the dorsal columns. And that, you can follow that branch all the way up the spinal cord into a region up here known as the brainstem. Now, if you take another neuroanatomy class or get into the weeds on this a little more, you're going to find that the brainstem is just a kind of elaborated, fancy spinal cord. If you end up looking at the anatomy of that structure, you're going to find that the brainstem is an extension of the spinal cord that deals mostly with the head. But for our purposes here today, I want to follow this process of the axon. The axon or conductor region has entered the white matter of the dorsal columns. If you put your phone away, I'd appreciate it. And then this process then ascends the cord and comes to this site here in a region in the brainstem known as the dorsal column nucleus. Now, again, this terminology, when we talk about a nucleus, we talk about a collection of neuronal cell bodies. You can read here input regions. In the central nervous system. So this primary sensory afferent synapses onto a second-order neuron. The second-order neuron that I'll show here in orange has its input region or neuronal cell body here in the dorsal column nucleus. This neuron then sends a process that crosses the midline in the brainstem and then continues to ascend until it reaches a section here in a portion of the diencephalon called the thalamus. The diencephalon, in fact, contains the thalamus and the hypothalamus. So now, if we follow the pattern that we have on the right-hand side of the board, primary sensory afferent comes into the central nervous system. Put your phone away, I'd appreciate it. Don't give me the look. You come in here a half hour late, you sit down and you pull out your phone. And then you give me the look like you shouldn't even be here. So I would appreciate it if you just get out. So thank you. The primary sensory afferent enters the spinal cord, ascends the cord in the dorsal columns, and forms a synapse onto the second-order neuron. The second-order neuron crosses the midline and ascends into a region called the thalamus, called the thalamic relay nucleus. In the thalamic relay nucleus, again, we have the term nucleus. That's a collection of neuronal cell bodies. In the thalamic relay nucleus, we now find that there are synapses between the second-order neuron and the third-order neuron, which now sends a process to a region called the cerebral cortex. Now, it's important to recognize here that what we have is a three-cell chain. Primary sensory afferent, second-order neuron, third-order neuron. The second-order neuron crosses the midline. The kind of information being carried here in this so-called dorsal column medial lemniscus. The dorsal column medial lemniscus is carrying information in this pathway, and it's carrying information about proprioception, where bodies are in space, and it's carrying information about what we call fine touch. And what will follow tomorrow is the other pathway, which is carrying information about pain, temperature, and pressure. And we'll see that they follow a same pattern: primary sensory afferent, second-order neuron, third-order neuron, to the cerebral cortex.